everyone. Good afternoon and welcome to the Protection Island Speaker Series. Today I'm happy to introduce Andrew Cameron, who many of you will have met over the last year here on the island. Andrew grew up in Nanaimo with a strong connection to Protection Island, as his dad, Norm Cameron, has lived here for many years. Andrew graduated with a BSc in biology from VIU. Then he went on to achieve a PhD in microbiology from UBC. Next, he did a postdoctorate at Trinity College in Dublin, Ireland. Andrew is a molecular microbiologist with a specialty in infectious diseases. He is associate professor in the Department of Biology at the University of Regina and is co-director of the Institute for Microbial Systems in Society at the University of Regina. Andrew studies the genetics of microorganisms and teaches multiple courses in genetics. He leads COVID-focused research projects to develop new genetic tools for detecting novel viruses and finding COVID co-infections. Andrew works in partnership with the BC, Alberta, Saskatchewan, and Manitoba Provincial Public Health Laboratories, as well as the National Microbiology Laboratory in Winnipeg. Andrew's an adjunct member of the biology faculty at VIU and collaborates with UBC, UVic, and SFU. Andrew and his wife, Jess, recently purchased a home on PI, and they will be residing here. Andrew will travel to Regina for his classes for 4 months of the year. Please use the chat box and or raise your hand, and there will be a question period at the conclusion. Thank you. Over to you, Andrew. And welcome.
Thank you very much for that introduction, Bev. And Emily, can you give me a thumbs up that you can hear me at least clear, if not loud, at Beacon House? Great. Okay, well, so nice to see you all, and thanks for having me back again, PI Lions. Last time I was here, I spoke about COVID and the genetics of the SARS-CoV-2 pandemic and vaccinations and things like that. Today's a bit different. Today, I'm going to be talking about CRISPR, which is a system that we do use in my lab, but it's not something that I research specifically. It is, however, something that I lecture on a fair amount in my multiple genetics classes because, well, it's one of those possibly most amazing genetic tools that any of us have witnessed being developed. And really it got its start or was proven to be effective at editing genes only 10 years ago. And in that 10 years, it has just rocketed in terms of expanded capabilities, what it can do, how it can manipulate genes, and different ways that scientists are adapting it to be able to change genes in adults or even babies. And we'll come right around to that at the very end of today's presentation. So I'm a biologist, and this is really, so I focus on the world of DNA sequence. And I see if my internet disconnects for any reason, just wait patiently for 2 minutes and I will pick up my computer and move closer to the router and be able to reconnect shortly. So some of the joys of Zoom in our sometimes weak internet world. Okay, right, so in terms of CRISPR-Cas, so this is a tool, and I'm going to give us an overview of the molecular biology. So what is the challenge that we're trying to address in developing this tool, and how does it get applied to do things like edit a human gene? What I'm not is an ethicist or a bioethicist, and I know many of you are probably most itching to talk about and think about the ethical implications of having such powerful tools for manipulating the biological world. And that's certainly where I want to end today, so that that's what segues into our question or discussion period. So I'll try be efficient at giving an overview of the genetics. And in putting this presentation together, I have sort of messed around with the order a little bit because I wanted to start with talking about CRISPR and what it is. But then I realized that it doesn't make much sense without having some basic human genetics. So I'm going to start with some human genetics to get us all on the same page and then talk about what is CRISPR. So with that, I'm going to share my screen. And again, I will look to Bev to confirm that you're seeing a full screen. It won't be a full screen yet. So Bev, you see the full screen and not my presenter
tools? Yes, now I can see a full screen. Yes.
Perfect. Okay, right, so there's me, and I can skip this slide because Bev's already given this introduction. I've also just talked a bit about how I'm a geneticist, and I've got a couple of slides here to give you just a little bit of intro to the research I do. Much of the time, I'm very interested in organisms and their relationship to each other and the world around them. And there's a new jargon term for that, which is really One Health, where we study the relationship between landscapes and people and animals and the bacteria that connect every living system on Earth. And specifically, I go after the genes to understand the complexities of these systems. So as you've heard, I've spent a long time in Saskatchewan, and there's me with my dog, Ginny, who many of you will have also met on Protection Island now, along with my wife, Jess. And I also have multiple projects ongoing in different parts of Africa, a major focus in Southern Africa, looking at infectious disease and connections between landscapes and human activities and animals and infectious disease. My lab works on pathogens in veterinary medicine as well on the prairies. And then we also work on zoonotic pathogens, so the types of things that we might get from a tick, so a tick-borne disease in, say, the forests of British Columbia. And then what I talked about last time I was here a year ago, or almost a year ago, was my work on molecular epidemiology. So using DNA sequencing to understand the spread of diseases and finding new genes and functions to understand those diseases. But today, right, is going to be much more a general discussion about genetics and the tools we can use to manipulate genetics. And that starts with just a little refresher on how we think about DNA and understand what this molecule does in our cells. And famously, the structure of DNA was solved really only about 20 years after DNA was determined to be the genetic material, so the molecule that carries the information that allows our cells to build bodies like each of us inhabit. And so there on the left-hand side, you will have probably seen these images of, this is Rosalind Franklin at the top, whose images were critical to solving the structure of DNA, which we now often see illustrated in figures like this, where it's a double strand of two separate molecules, actually, each gray line, and they're connected together by base pairs. And as a geneticist, I think about the DNA certainly in that structure, the sequence and the information that can be read from that DNA sequence. I also spend a lot of time thinking about the structure like this, and that's essentially the same DNA molecule, but now shown with a little more space-filling molecular structure. And the shape of all those bumps and ridges is important to how the cell uses DNA. So what are the transactions that the cell can do with its DNA and read that information? And that's going to be sort of a key theme today. So to wrap up about me and my interests in these types of systems, as a molecular geneticist, I would seek to understand things like how information is stored in DNA and how do cells access that information to make usually the proteins that the cells need for their function. The consequences of changing the sequence. So things we often refer to as mutation. Won't touch on that specifically today, but I do have some neat slides in case we come around to discussions about different types of mutations. Tracking mutations, a great way to understand infectious diseases as an example. And then I'm also very interested in how genes evolve. So a lot of the research in my lab is also evolutionary questions about how genes change over time to adapt to different environments. So in today's presentation, I'm going to start with this intro to human genetics and then talk about disease, then to the overview of what is CRISPR-Cas. And I should refer to it as CRISPR-Cas, but as so many of us do, I'm just going to truncate the name to CRISPR, which is completely technically incorrect, and I'll show you why in a few slides. Okay, so how much DNA in a person? This is our first thing to think about today. And in that illustration of a DNA molecule that you see on the left-hand side there, there's 16 base pairs, right? So enough base pairs that I could count them on 2 hands and 1 foot of toes. Now, how many of those base pairs are there in a human genome? Well, each human cell, with a few exceptions, contains over 3 billion bases that are inherited from mom and 3 billion bases that are inherited from dad. So in total, each cell in our body contains approximately, or just over 6 billion base pairs of information. And collectively, that's what we call the genome. So the genome is the term for all of that genetic sequence in any one of the cells in our body. And as we know, the genetic sequence is essentially identical in all the cells in our body, again, with a few little exceptions. And just to show you a figure that you might be more familiar with, if you're remembering back maybe to some high school biology or intro biology classes, is that all of that DNA, that 6 billion base pairs, is packaged and stored into these large structures that are chromosomes. And that's the image that you see at the upper hand side there. One of those chromosomes came from mom and the other one was inherited from dad. And that's how we get of each of the chromosomes in each of the cells in our body, which is a good thing because that provides a lot of buffering, for example. And I won't get in mind as we talk about mutations is broken. Quite often, the functional copy can compensate and make the, say, proteins that are required for the cell to function. And please do type any questions in the chat, but I'll just keep on going here. So now let's think about mutations and the differences. So essentially the variants in humans. We hear the term variants when referring to coronavirus all the time now. It's become part of our standard language, but humans are just the same, right? We've got many, many variants, and I'm going to walk us through the numbers to try to imagine how many different genetic variants there are in humans around the world. So if, for example, here's some sequence of some DNA, I'll just say in my cells, and I got one copy from my mom and one copy from my dad. Now, those sequences in my cells are doubled because they're base paired, but for convenience in genetics, we always just write one single strand because it's easier to read. So that's why you see 2 lines of text there, one from my mom, one from my dad. Now, those 2 lines of text are identical, except at one position there, where there's one base difference between the copy I got from my mom and the one I got from my dad. And I'll talk about the implications of that precise mutation over the next few slides. But what that does in this example is that changes the protein of the gene that piece of DNA codes for, right? So that DNA information to make protein in my hand, the copy makes a protein at that position, whereas the copy from my dad puts a valine at that position. Now, quite often that doesn't have any implications, but in this precise case, it does have a major implication for health, and I'll talk about that in a couple of slides. So just for some gene capture, I've sent your DNA off to phenotypes or features about our body that are encoded or differ based on a single base character in the DNA that we get from mom and the DNA we get from dad. So here, this 23andMe is calculating what's the probability, if you know the genetics of mom and dad, that what will their offspring look like.
Andrew, the sound is breaking up.
Yes.
Is everyone having this trouble?
Yes. So as I was saying, some of the fun of genetic determinants. So here we see a sequence illustrated by 23andMe, where single base changes, and each parent, of course, has 2 copies of each position. And we can see things like bitter taste perception and the probability that the offspring of these 2 parents will be a non-bitter taster because that child will inherit a C from one parent and a C from the other. So these are the types of, well, fun little things that we might not necessarily want to go in and edit to try to fix a person from a non-bitter taster to a bitter taster. But exactly this type of genetic difference is exactly what CRISPR is particularly well suited to manipulate. Okay, so how much are we different from one another? Any two people in the human population, we could say on average, differ by about 6 million base pairs, right? So we all have the same positions, but at each one of those positions, we might have a different sequence compared to another person sitting beside us, say today. How many variants do we recognize in the whole human population? So if we look at all of those genetic differences, it's probably well over 150 million, because as we sequence more and more human genomes, we start to see more and more diversity at these different base positions in the human genome. And then finally, how much do we differ from our parents? So we get perfect copies essentially from each parent of one copy of their genes. But then as we grow and divide, right, you're going from a single cell, I'll come back to that, to a body that's composed of trillions of cells. Errors happen as the DNA is being copied. And so we end up with maybe 50 to 150 base positions that are different between us and our parents, just due to mutations arising during our normal lives. So on average, then, people differ from each other by about 0.1% of their sequence. So it's about one base pair out of every 1,000 base pairs. You may be different from the person sitting next to you. So I'm just opening up my chat here so I can see too many things going on in my little window. Okay. So now let's think about, we've got so many bases in our genomes. They're organized into chromosomes, and along the top here, I'm showing chromosome number 11. And then I'm zeroing in on one position on the left-hand side of that chromosome. And I'm zeroing in on this gene called HBB. So that's what you see highlighted in green there. This is a relatively small gene for a human gene, and it codes for just a small number of amino acids to make a small protein. This is a very, very important gene. It makes hemoglobin, one of the types of hemoglobin that our cells use. So if we zero in more on that gene, here's that sequence I showed you earlier. So now I'm going to show you on the next slide, zooming right in on that sequence where between my mom and my dad, and again, this is a hypothetical, that I have two different versions of this gene in my cells. So if we zoom into that gene at the top, and now I'm going to show you about 50 base pairs of sequence from that gene. That's what's in gray along the top there. Now I'm going to show you different mutations that we recognize across humans around the world at this 50 base pair position in this hemoglobin gene. So there's many, many, many different genetic variants. And for each one of those red dots, for example, that's a different position where there's a mutation in some human out there somewhere who's had their genome sequenced. And in addition, at any one of those positions, it could be mutant into any one of the different nucleotides. So that's what we also see indicated there as well. So each red dot could actually represent multiple types of different mutational changes. Along the top, those purple bars show where there's actually large rearrangements in the gene. So often deletions, deleting a large section of a gene is not uncommon in our genetic systems. Now, what's amazing, right, is that a person can lose a chunk of their hemoglobin gene and still continue to make blood and be a living person, or we never would have been able to sample their DNA and sequence it to see these types of mutations. And that's because we have two copies of each gene, right, that we can tolerate some of these large types of mutations. But today I'm going to focus mostly just on those single changes, and I've highlighted that one there in the little yellow box that just popped up. I don't know if you can see it there, Beacon House, but that's the hypothetical mutation that I said I might have, where I've got one version from mom and one version from dad. And that's, oh, I'll tell you in a second what that causes, but most of these mutations really have no biological significance, right? They're just diversity in human genetics. They don't have benefits that we recognize. They don't have negative consequences necessarily that we recognize, except for that base that I've highlighted there in gold, where that change can result in a very serious disease. And that's the mutation that can cause sickle cell anemia. So it's just a single base pair change from glutamic acid, which would be considered normal, which most people on Earth have, and then changing it to a valine at that position changes the structure of the hemoglobin, which is stored in red blood cells, and that consequently changes the shape of the red blood cells, which can result in a disease state. Okay, so let's talk about if we want to address and cure genetic diseases, of which there's many, many thousands, where we know of mutations already that contribute to those diseases. That's a very complex topic. So again, we can explore that in later discussions. But there's many technical challenges if we want to correct DNA sequence in a person to try to cure a genetic disease. So first off, we need to find that single base pair that might be causing that disease state out of over 6 billion bases, right? So that's really a needle in a haystack. And then we want to change that base pair to a very specific alternative, because we don't want to create additional mutations and potential problems for the person. We want to correct that sort of mutant form back into maybe the healthy or normal form. We do not want to cause other mutations in other genes while we're doing this process. So that's no small consideration as well to manipulating genes. And for many diseases, we might need to change millions or billions or even trillions of cells in the human body to actually correct that disease. So that's another big challenge. And I'll touch on each of these over the next few minutes. Okay, so now about CRISPR-Cas. What is it? Well, at its simplest, it's an enzyme, but we can think of it as a pair of scissors that is able to cut DNA at a very precise location. So that's what CRISPR-Cas is a very targeted pair of scissors to cut DNA. Sorry, I'm having trouble seeing all my screen with my little laptop view here. So some of the shortcomings of early efforts to do things like this, to go in and find a base pair and edit it, were that scientists had to use proteins to do that, because it's proteins that are the machines in cells that fix and manipulate DNA. But one big problem is that proteins feel their way along the DNA. So remember that bumpy space-filling model of DNA I showed you? You can imagine a protein, which is a very different kind of molecule, feeling its way along, looking for the right sequence. Most of the time, the proteins can find the right sequence, but they also find a lot of incorrect sequences that are very, very similar. So there's a risk of it being inaccurate, this type of approach. And also, anytime we want to fix a different base pair somewhere in a different person, or even in the same person, we would have to engineer a whole new protein structure to go looking for that other DNA sequence. And that's really no small feat. That's very, very laborious to do. So this is where CRISPR-Cas has changed everything. So the great innovation of CRISPR-Cas is that it's a protein, but what it does is it grabs an RNA molecule, and I'll show you this on the next slide, and RNA is able to perfectly base pair with DNA. So now CRISPR-Cas is using like to find like. It's using that RNA molecule to look for the precise sequence in the DNA where it's going to make that cut. It has been discovered that all of the functions of hunting down that base and causing a cut can be engineered into a single protein. So that's also really efficient, and I'll maybe talk a bit more about that later. And we've also learned that we can attach other enzymatic functions to this targeting mechanism that CRISPR-Cas is so very, very good at. So highly precise, very easy to engineer, and it's now a very versatile toolkit because we can add other functions to it. And here's what it looks like. And I won't go into too much detail about this molecule and this image, but in pink there, you see a DNA molecule. And in this green globular form is the Cas protein. So this is what we refer to as CRISPR. And it's carrying this blue molecule that's the RNA molecule. And what it does is it scans along the DNA molecule looking for precise base pairs to this RNA molecule. Wow. And that's what makes it so precise. And I don't have an image to show once it's achieved all that pairing to know that it's in exactly the right location. But once it does that, if you can imagine that blue molecule base paired precisely to the pink molecule, then that triggers this Cas protein to cut the DNA It engages its scissor activity and cuts the DNA at that very precise location. Does that make sense? So that's really CRISPR-Cas. That's what it does. Wow. And it does it very, very well. Okay, so what's in a name? Why do we call it this very awkward CRISPR? And CRISPR is an acronym, and it stands for Clustered, Regularly Interspaced, Short Palindromic Repeats. And why I like its full name is that it tells us something very interesting about how CRISPR was discovered. So CRISPR was discovered really way back in the early 90s when bacterial genome sequences were starting to become available. And in the early 90s, there was only a handful of bacterial genomes where we had a fair amount of sequence. Nevertheless, even with just that little bit of genetic information, scientists were able to notice that there were these clusters of repetitive sequence. And so they called it CRISPR. And henceforth, we just say CRISPR instead of spelling out the full name. Now, what that tells us also is what I said earlier about CRISPR not being the right name for what we're all talking about with this DNA editing function, because that term refers to finding these repetitive elements in a genome. And I won't go into this image in too much detail, but this gives you a sense of what CRISPR looks like in its natural form in a bacterium, where those black diamonds are the regularly interspaced repetitive regions. So they're the repeats. And what geneticists noticed was that there was a whole bunch of these diamonds spaced at always exactly the same spacing from each other in an array in bacterial genomes. So why? What is that? Oh, but first I'm going to talk about Cas. So more appropriately, we should refer to the whole system as CRISPR-Cas, because it's actually the Cas protein or proteins that we use to do genetic engineering. And Cas just stands for CRISPR associated. And in this example here along the top, I should have said, sorry, is my DNA molecule, where there's this array of the CRISPR elements. And just adjacent to that are genes that are these white boxes here that encode the enzymes that I just showed you on the last slide. So the enzymes that will bind the targeting RNA and have the scissor activity to go cut DNA. And that's the whole system. Oh, and Lynn, I see your question and I will come back to it. Okay, so a little quick note on Canadian connection, and Canadian researchers have contributed immensely to understanding CRISPR, and I'll just go back a second to simplify this. And I think this is a fun little story because it has to do with cheese. So what do cheese and CRISPR have that connects the two of them? Well, as you can see from those numbers there, cheese is a big deal in Canada. We spend over $4 billion a year, Canadians, on cheese, and that number probably is just getting bigger and bigger very, very quickly these days. And one problem that cheesemakers have is that when they're taking those hundreds and hundreds of thousands of liters of milk product to make cheese, they use bacteria to do that. And there's one bacterium in particular, and that's shown here, Streptococcus thermophilus, that is used to make things like yogurt and cheese. Now, the big problem that cheese producers have is when they take those hundreds of thousands of liters of milk, put it in a vat and mix in the bacterium, they hope that the bacterium is going to do the right job and metabolize that milk to make a cheese product. What happens if you put your bacterium in and then it dies? Well, now you could lose hundreds of thousands or millions of liters of milk, and it's devastating to your industry. And that's exactly what Canadians were studying, was that they found how the bacteria in cheese production can become resistant to viral outbreaks. So there are viruses that will infect and kill bacteria, and those viruses are devastating to the cheese industry. So what's happening here, and what the Canadian researchers discovered, was that this CRISPR-Cas system is in fact an immune system for bacteria. And it works like this, that on the left here, we see the virus, and we call them phage, the ones that infect bacteria, but they're viruses. When the virus binds to a bacterial cell, it injects its genetic material, in this case, a double-stranded DNA molecule. But in some cases, or many cases, the bacterium doesn't actually get sick. It's able to defend itself against the virus, and it uses CRISPR-Cas to do that. And here's the Cas protein that I showed you earlier in that sort of blue-green space filling model where it's got its guide RNA, and its guide RNA targets it to destroy the incoming genetic material from the virus. And so the bacterium defends itself against the virus by cutting up the viral genetic material so the virus can't cause an infection. And understanding that dynamic has actually helped cheese producers, but cheese producers more than anything have helped understand CRISPR in its natural context in the biological world. Okay, so new versions of CRISPR are coming into play all the time. And this is a slightly complex slide as well, as are the next few, but I'm going to walk you through them just to get the sense of how this system works. And when I'm describing the scissors, and this goes specifically to Lynn's question about if the enzyme is just a pair of scissors and it cuts the DNA, Lynn, you're totally correct that if you cut the DNA, it's actually really damaging to the cell. And the cell has many repair mechanisms that it throws into action to repair that cut DNA. Unfortunately, that results in additional mutations often. And so CRISPR-Cas can be very good at targeting the right location, but sometimes it can make a mess of where it's made that cut. And so the great innovation was to get rid of that double DNA strand cutting mechanism and refine it so that CRISPR-Cas maintains its precise targeting, but now it introduces just the specific mutation that the scientist wants and does not cause additional problems where that cut has occurred. And so in this new precision editor version of CRISPR, here we have the DNA strand that the scientist wants to repair. They want to switch that X sequence with the Y sequence. Now CRISPR comes in, it uses that RNA to target, knows that it's at the right location, but now instead of cutting the whole DNA, it just replaces the sequence on one strand, gets rid of the little piece that it's displaced. So now we've got a mismatch where our double-stranded DNA molecule has two different bases that don't necessarily prepare with each other. So how does the enzyme then repair that second strand of the DNA molecule? It now creates just a little nick on the other strand, and the cellular machinery comes around and copies to introduce that mutation now on the other DNA strand. And now the resulting DNA strand, this is one enzyme, that Cas protein has done all of these functions and has completely swapped out that DNA molecule with a new sequence. So this is highly precise. We don't expect it to make mutations in the other 6 billion bases in the cell, and it's very clean. Okay, so now let's talk about diseases to really wrap up here and talk about how can we use this tool to fix genetic diseases. Well, the thing that's probably most heralded and most exciting about CRISPR-Cas is what I've just described, this ability to take a disease like maybe sickle cell that is encoded by a single base change, and then being able to use the enzyme to go in there, target, and change that base. And that's what's illustrated in this cartoon at the bottom here. There's the Cas protein going in and cutting and changing that sequence. Also very exciting is that we have infectious diseases that cannot be treated with any type of medicine when they are integrated into our genomes. And so key examples of that would be things like the human immunodeficiency virus, HIV, that causes AIDS, and herpes viruses, that these viruses, in addition to their normal life cycle, part of what makes them so hard to fight is that they can actually integrate into our DNA. And once they become sequenced in our genome, our cells are no longer infected in the classic sense. They're not producing virus at the time. So our immune systems can't detect that the cell is carrying the HIV genome, for example, and then that HIV can emerge at a later date. And so we see things like this with cold sores and herpes, for example, is that reemergence of the virus that's being encoded by your cells that's impossible for your immune system to see. Now, CRISPR-Cas is very attractive now because by targeting the CRISPR enzyme, it can see the HIV that's hiding out in the cell's genetic material, because we can target CRISPR by giving it the RNA molecule that matches the HIV sequence. CRISPR goes in and finds that hidden HIV and cuts it and destroys it. So that is very exciting, because now that's the first time that we would actually have a tool to essentially remove a virus that's hiding in a way that we can otherwise not treat with vaccines or medication, right? But now the really daunting challenge that HIV in this example, or any disease-causing mutation, is not just in one cell, right? It's in many, many different cells. So we have to look at repairing all those cells. And I'll touch on that in the next slide. But first, a few more things about this, this CRISPR system and some of its powers, I guess. I was going to say benefits, but one is that in a lot of ways, it can circumvent at least some of the regulations around genetically modified organisms. So for better or for worse, in discussing genetically modified organisms, that's a whole other discussion. But what many nations are currently allowing is for CRISPR-Cas manipulation of organisms is not considered genetically modifying those organisms. So yes, you're changing the gene content. You can be introducing all sorts of gene functions that you want, but because the CRISPR itself does not persist in the organism, it goes away when we're done using the tool, we don't consider, or many don't consider that organism to be the classic genetically modified organism. The EU is more stringent about this, but places like Canada at the moment would not consider CRISPR modifications to be GMO. Some of the real benefits, as I've discussed already, is that it can do more than just cutting, right? So it can do all sorts of things now that we have such a precise way of targeting different DNA sequence. If we remove the cutting function, we can actually have Cas deliver other enzymatic functions to very specific genes and locations in our genome. And we can adapt it to work in almost any organism, right? So in my lab, we use CRISPR to make mutations very quick and efficiently in different bacterial species, like Salmonella, for example, that we're studying. So it can be used in humans, it can be used in other mammals and other animals. It's used a lot now in plants to modify those plants. Okay, so how do we get it into our bodies? And again, I unfortunately don't see the time because my screen is so small here, but I've only got 3 more slides, I think, to really wrap this up. Okay, so how do we get it into our bodies to make these genetic changes? And in the center of your screen here, you see the CRISPR enzyme is that golden blob. And here, this DNA sequence is the gene sequence that, say, the scientist or clinician wants to change in the patient. So how do you get that enzyme and the corrector, the correcting gene or sequence into the person's cells? Well, one way to do it is to put those tools into viral particles and then allow those viruses to do all the work. So the viruses will deliver the CRISPR-Cas gene system to the cells, and then the cells will read the genetic information. They'll make the CRISPR enzyme or CRISPR-Cas enzyme. They'll read that, in this case, therapeutic gene, as I've labeled it, and correct the change or whatever mutation is being targeted in the human cells. So what you see on the right-hand side there, that injection of the viral vectors. This is also quite similar to vaccine delivery in even some of the COVID vaccines, like the AstraZeneca is using a virus to deliver the genetic material into our cells. Now, a very similar approach is to take that genetic material and put it into little nanoparticles. So these are little lipid particles that can carry, again, the genetic material inside that codes for CRISPR-Cas and the therapeutic gene. And this is akin to what those of us who have received an mRNA vaccine for COVID, this is how it was also transported into our cells, where the genetic material to make the spike protein against COVID was put into one of these little non-viral nanoparticles, this little lipid carrier that can take the genetic material inside. So these are very localized treatments, right? Where we can inject CRISPR-Cas into target organs. And I'm going to tell you about a couple of successes in a couple of slides. And sorry, Lynn, I see there's. Okay, and I'll adjust the COVID question at the end. Now, probably this would have been, or this was the first way that CRISPR was used effectively in study systems in rats and mice, and now in humans as well, where instead of injecting it right into a person, instead you can take some tissue out of a person. So let's say some bone marrow, for example. So if a person is producing sickle cell proteins, has that hemoglobin mutation. If we take some bone marrow out of their bones, which is what's responsible for making the red blood cells, and use CRISPR, and this busy slide is just showing that once you've taken the cells out of the person, you can introduce the CRISPR-Cas system through injecting the DNA right into the cell. You can use the viral vectors or the non-viral delivery, as we've just seen on the previous slide, to now infect these cells that are in culture in the lab. And once you confirm that those cells have the genetic change that was intended by engineering them with CRISPR-Cas, you can actually put those cells back into the patient. Whoops. And once you put them back into the patient, now those cells are able to grow and proliferate and produce the corrected gene. They'll express the corrected gene. And in my example, they'll produce the correct hemoglobin molecule. And that person's blood now will be replaced with the proper functioning hemoglobin. I'll address the Down syndrome question in a couple more slides as well. So just to wrap up now, some recent successes in using this tool to change genetics. Oops. Sorry, this is my problem with too many slides here, or too many things open in my little window. In mice, of course, where these tools were first shown to be functional. And just as a quick aside, in 2020, the Nobel Prize was awarded for the discovery of CRISPR-Cas. It wasn't really the discovery of the tool, it was instead showing that CRISPR-Cas could be used to manipulate mouse genes very, very effectively. And the two scientists who really led that research are who was awarded the Nobel Prize, and they did that research back in 2012 and won the Nobel Prize for it in 2020. So in mice, there's been many, many advances, and multiple diseases now have been shown can be cured, because in mice, you can change the genes through the processes that I just illustrated. And you can also make CRISPR babies in mice. And I'll talk about CRISPR babies on the next slide. So yes, there's already been shown to solve different blood disorders in mice, and Huntington's disease are examples. Now, a very recent example in humans where CRISPR has been used directly in adults, actually injected, as I illustrated on the previous slides. So this example here, this transthyretin amyloidosis, is a fairly rare disease, but a very serious disease. And up until very, very recently, there really wasn't a treatment for it. So the problem in this disease was a misfolding of a protein called TTR that causes fibers to build up in organs. And it mostly impacts nervous tissue and can impact the heart. The liver can destroy their functions. So you can see heart disease as a common outcome, very painful, and can cause death. And there really was no medicine to fix this, this disease. So what has been shown now in a clinical trial that has had great success was 6 individuals with this disease were treated with CRISPR-Cas. And it was using an older version of CRISPR-Cas, so not this new high precision editing tool, but just using its old cutting functions, where the scientists injected Cas9, the guide RNA, to tell that Cas9 where to go to target the TTR gene. And then it went in and it cut and essentially destroyed that misfunctioning gene. So it knocked out the mutant form that was dysfunctional in these people, and they then lost that fiber production and were able to recover from this disease. Okay, so finally, the big one that creates probably the most concern from an ethical perspective is when we have a tool that is so easy to work with, and I haven't stressed that enough, but the components, so having the gene to make that Cas protein that does the targeting and the cutting, making the guide RNA that tells it where to go, are incredibly simple to do. And you could literally work with this tool in your garage if you just had a few simple home tools to mix some of those components together. So very easy to manipulate. And as a consequence, right, this is a powerful tool now that immediately people recognize could be used to edit human babies, essentially. And why is this so powerful? Well, all of us at one point in our lives, we were a single cell, right? So when our parents' sperm and egg fused, we then became a zygote. So we were one cell big at that time. And that's, if you make any genetic change in a zygote, then that genetic change will be passed on to every single cell in the human body as the human grows and develops. So that's why this tool is so powerful, that if you want to introduce new genes, change gene sequence in the ways that I've described, if you do it in the zygote stage, it will change that whole human as they grow and develop. Now, one of the major concerns, at least from what we know about the biology and how this system works, is that although CRISPR-Cas is very, very precise, and we know exactly which gene we're going to manipulate, and we can be quite confident that there won't be lots of off-target mutations, one big problem is that we don't know what the consequence of changing that gene is going to be. So could it alter the development of that child by changing that gene? So maybe we know that mutation causes a disease in adults. Maybe that mutation actually does something very different early on in development, and we don't know that until we've experimented and done this on a human, essentially. It could be incompatible with other genetic variations in that individual, right? So although we might make a change that we see is common in the human population, it might not actually fit with the genetic makeup of that individual. So these are some of the big problems, is that we could create new diseases and new developmental problems by having such a powerful tool to manipulate at the zygotic level here before a human has even started to grow and develop. Okay, and now I will address a couple of the questions in the chat, and then Emily, I'll pass it over to you to moderate, or Bev, a question period. But there was a question about how does CRISPR find the new sequence that's to be inserted? And so I've described how there's the enzyme that takes that guide RNA, and the guide RNA targets it to where we want the mutation. And then you're quite right in that question that there's now a separate piece of genetic material. And several slides ago, I called that the therapeutic gene. That also gets injected along with the Cas protein and the guide RNA. So there's really three ingredients. They're all quite simple and small and easy to manipulate. Is long COVID due to COVID-19 getting into and binding to our DNA? No. So some viruses, and particularly some RNA viruses like HIV, are able to integrate into our DNA. SARS-CoV-2 that causes COVID-19 does not do an integration function like that. That's not to say the virus itself can't hide out in your body for long periods of time, because another problem or a challenge for our bodies when we get infected with diseases is that although our immune system can be very, very effective at finding all the COVID and destroying it, there are parts of our body, like our reproductive tissues and also our nervous system, where our immune system isn't really allowed to go. Either it's not allowed to go, or it's only allowed to have very limited influence. And that's because our immune system can be quite damaging. So there's parts of our body where our immune system just isn't very active. If the virus gets into some of those locations, now it can persist for a long period of time and still cause reemergent infection, even though it's not integrated into our genetic material. Long COVID, more specifically, and I won't go off too much about COVID, but another aspect of an infectious disease like COVID is that it results in tissue damage, right? So we get a lot of inflammation and destruction of tissues that will mostly be in the respiratory tract in something like COVID. And that tissue damage can create long-term negative impacts. There can be other tissues that get damaged due to the immune response against that infectious organism. So another very good reason that vaccines are so safe is that they greatly reduce the chance of those long-term or very negative consequences of getting the real infection. So what controls are there for the use of CRISPR-Cas in engineering humans? Well, There are some formal laws, particularly advanced in places like the EU. Again, I will be careful not to misspeak, but I don't know a very good survey of all the different laws that are being considered and introduced in different parts of the world. But often legal regulation of systems like this lags very, very, very far behind the development of the tools themselves. So often there can be things like just essentially societal agreements that we will not make CRISPR babies in Canada, even though there's not necessarily a formal law on the books yet that prevents us from doing it. There will be an agreement among the scientific community that there is too much unknown about the consequences of using this tool. So we need to keep studying this tool and essentially go after the low-hanging fruit, the diseases that are most readily treatable without manipulating zygotes before they even become babies and grow and develop. So that's a tricky question, and we certainly do know of places on Earth, and including scientists in China who have been public about their manipulation of human zygotes to change human genes in essentially new humans before they've grown and developed. Okay, those are the questions I see in the chat. I will stop sharing. Are there, if you have any questions, please just raise a hand or put it in the chat. Sorry, Bev.
I was just going to say, are there any more questions for Andrew? Anyone out there?
I see some at Beacon House, some hands up.
Oh yeah, I can't see that, so let's go there.
Hang on, Bev. Hang on.
Okay.
Okay, I can hear you, Beacon House.
Yes. So, Peter, can you have the people at Beacon House ask their questions?
Right, and then Bruce.
All right, if you want to ask your question, maybe
come and talk.
Yeah, the microphone I think is up there.
Is it? Can you hear me?
I can hear you, yes.
Okay, Andrew, you, right at the beginning you said that in a human there are about 100 base pair mutations that occur during lifetime, correct? How many of those are heritable, or are they all just somatic mutations?
Yes, so whether you can pass any one of those mutations that happens as you grow and develop, which are just often due to mistakes, as to make a trillion cells, you have to copy your genetic material many, many, many times, right? And so that's where these little errors, these little mutations arise. Can you pass them on to the next generation? So if they happen very early in development, yes, you can, but, and this is probably very deliberate in a biological and evolutionary sense, the tissues that go on to make our gametes, so produce our sperm and our eggs that will go on to produce the next generation, they become very, very, very early on in our development, they become dedicated tissues that are, have, in some cases, lower reproduction rates. That's not true for sperm production, of course. Men are responsible for most mutations because of having to copy so much DNA to make sperm. So many of the 100 that I described that, so let's say that you wanted to have your genome sequenced and you gave a swab from your cheek cells that was sequenced, and comparing to your parents' genomes, we could see about 100 base pair differences. Most of those would not be in your germlines. You would not be able to pass them on to the next generation. The types of mutations, as I commented, there are many types of mutations. There's deletions, there's large reorderings, so pieces of chromosomes can move to the wrong place. Unfortunately, a lot of those mutations can arise in our germlines, so the tissues that are going to produce our sperm and eggs, and then we can pass those on to the next generation.
Thanks.
Bruce, can you hear me from here?
Yes, I can hear you.
This is half a joke, but in video games, there are things called map editors and things. So is there any evidence, is the existence of CRISPR some evidence that maybe we do live in a simulation?
Well, I mean, this goes back to the point that, and I don't know if I was able to capture this well today, but CRISPR is present in probably most of the living things on Earth. It's just they're bacteria. It's not part of our human genetic system. And it has a very clear function in those bacteria that is not to do with this type of manipulation. So I would think from that evidence that that's very unlikely that the video game we may live in hid that CRISPR tool in those bacterial genomes. So it really is a tool that we have discovered in nature, like pretty much all of our best biological tools, things like antibiotics, we discover them in nature. And CRISPR is exactly the same, where we discovered an enzyme that had the right features that we could then adapt to use in a very targeted way to manipulate human genetics. So Lynn asked, oh, I will come back to the Down syndrome question. How does one go about getting their genome sequenced? Well, that has been the sort of holy grail of DNA sequencing since the launch of the Human Genome Project back in the 90s. And the billions of dollars that were invested into DNA sequencing technology to make the Human Genome Project possible and a success has resulted in rapid and massive advances in DNA sequencing. So whereas it cost hundreds of millions of dollars to sequence the first human genomes, and they were published back in the year 2000, nowadays it does literally cost less than $1,000 to sequence a human genome. And that's always been the goal, is get it under $1,000. And then it's thought that most wealthy parts of the world could afford whole genome sequencing as a tool to use in the clinic to look for genetic diseases. So the cost is now not really prohibitive. The technology to actually sequence a person's genome is quite readily available, not to the point that it's available in every clinic yet, but it's not too far off. The major challenge still is that, and if you think back to my slide showing you the hemoglobin gene, and what I wanted to capture there was that there's one mutation we care a lot about because it causes sickle cell disease. When we look at human genetics in their entirety, we see that humans can have mutations or differences, variants, I should say, all over the place. Even at the same position, they can have different sequence that doesn't cause sickle cell. So actually making sense of all of that, hundreds of millions of different variants in humans, that's really the limitation at this point, is drawing the connections between, aha, I see this sequence and this sequence and this sequence in individual A that I just sequenced their genome. How do I now make sense of it and think about, okay, well, I could use this type of medication, or maybe because they have this genetic anomaly, CRISPR could be used to go in and fix that. So yeah, sequencing a whole genome is, yeah, it's a whole other discussion too, Lynn, that I'm happy to follow up on, that when you send your DNA in, say to 23andMe or any of these genotyping places, they're not sequencing your whole genome. What they're doing is they're just sequencing many of those, about 6 million plus locations where we know humans differ. So they don't have to sequence the whole 6 billion bases and then try to put it all together. So that's much more efficient and much more cost effective in many ways. But sequencing the whole genome and making sense of it, that's definitely where the technology and the desire is going, because we will continue to learn a lot. And we already have, and I haven't captured that here, but even having the human genome sequence has revolutionized so many aspects of medicine. I don't think we hear much about it, and it's not really celebrated that much. We tend to fixate more on, well, when am I going to get my genome sequenced? But it really has been revolutionary for medicine. And we didn't anticipate CRISPR when the human genome was sequenced. But now that we have CRISPR, starting in about 2012 or so, and starting to use it in humans a few years later, having the human genome sequence is really a prerequisite for being able to use CRISPR in a targeted and effective way, because we need to know what are all 6 billion base sequences so that we can target CRISPR to just the right place, to not mess around with all the other bases that we don't want it to cut or manipulate. And so Down syndrome. So yes, I've focused primarily on single base changes, and most of the genetic variation in humans is those single base differences at a location where it may be an A or a T or a G or a C, right? So most of our genetic diversity is those types of differences. But as Lynn is alluding to with her question about Down syndrome, there's many other types of genetic mutations and anomalies that can arise. And sometimes pieces of our chromosomes can be copied, duplicated, triplicated. They can be attached to the wrong places, because every time our cells divide, one thing that they do is they align our genes and essentially shuffle them a bit. So that's essentially, the cell needs to do that to know that it's separating chromosomes correctly to daughter cells. And as a consequence of those other types of genetic transactions that are happening in a cell, there can be these more major reorganizations. And Down syndrome would be an example of that, where there's inappropriate number of copies of certain genes that then results in that disease state. So can CRISPR correct those genetic anomalies? Quite probably yes, because CRISPR is being developed so rapidly to do more than just change single base pairs. And that I described the simplest form of change those single base pairs. But by having an enzyme that can target so precisely, there will be very cunning ways that scientists will come up with to alter much more major genetic changes like occurs in Down syndrome. So not yet, but I would not be surprised if such things were just around the corner, really, as a way to manipulate human genetics. And then I think there was a question about how do you get it to all the cells? Well, that's where the issue around editing at the zygotic stage versus editing in the adult is, it's very attractive to think about, where we can recognize some genetic disorders and see them in the zygote before the human has started to develop and grow into even 2 cells or more. If we can make the changes then yes, you can correct all the eventual cells in the adult. Changing all the cells in a fully formed adult human, probably impossible. But there are some viruses that are quite good at going to multiple different tissues. So that is a very active area of research, using these now synthetic different lipid carriers. It will give ways for scientists also to develop ways to target CRISPR to deliver to multiple different tissues, but it's always going to be down to ultimately some random chance that you hope that you transform enough cells to correct whatever the organ or whatever dysfunction is happening as a consequence of that genetic anomaly, and then be able to have the body ultimately rely on the repaired cells, or those that have had the correction brought about by CRISPR, to now take over and produce the correct protein. So it's tricky, but again, this technology, right, 10 years old, and already it's being used to treat serious human diseases and effectively in adult humans, is really just shows how easy it is to work with and how powerful it is as a tool. Diabetes, yes, different forms of diabetes, certainly those with genetic determinants. Again, really anything that we can find in our DNA that we either predisposes us, puts us at higher likelihood of different diseases, will ultimately be something that can be modified by CRISPR-Cas. One thing I haven't said is that most of our genetic diseases are actually the combination of effects of multiple genes, right? So sickle cell anemia, where it's a single base change, is great for discussions like this or teaching purposes, because it's one base change creates that disease state. But for most aspects of our body, right, it's multiple genes working together. And so you can have multiple mutations and genetic variants in different locations in the genome that need to be considered and maybe manipulated in their entirety to correct a genetic disease.
Rick has a question at Beacon House.
Thanks, Beth. I hope you can hear me, Andrew.
I can hear you, yes.
Great. So it seems like the mechanics of CRISPR is pretty straightforward. So I'm just wondering, like, where does the science part of it fit in, and everything else other than CRISPR seems to have to go through a whole bunch of series of trials, and it might take years or decades to get to humans. By gene editing with CRISPR, like, the first part of it is the science, and then the second part is like the time frame for getting approval to actually try it on humans. Is that much faster then?
In terms of getting approval for clinical trials, I really can't say, except that it's probably fairly slow. So just judging by the currently limited number of cases where CRISPR has been used in humans to treat specific diseases, those cases have been very small in number. So another big one that was announced in 2020 was correcting an eye disorder in a single adult, where it was only a single adult in the whole trial. The fact that those successes, which have come in 2020 and 2021, we're using an old version of CRISPR. So they're often using that old version that just does the cutting, the highly targeted cutting, I think speaks to how long it takes to develop something that can get that approval to be injected into a person to test a clinical outcome. Severe diseases often change the calculus on clinical trials. So when we have diseases that are very acute and a person might be suffering, and if they don't get a cure sort of within weeks or months, then that can accelerate or lower the barriers to trying some of these tools, right? So those experimental technologies like these types of gene editing tools, if that individual knows that their alternative is that they're going to die possibly a painful death from whatever that disease is, then that can accelerate the allowance of being able to inject them with something still experimental like CRISPR. Because even though, and I think this sort of speaks to your point, Rick, that CRISPR the science is very advanced and that it's very easy to manipulate, but any given mutation or set of mutations that gets introduced to correct something like an eye disorder or different types of blood disorders, which also have advanced to clinical stages in humans. Each one of those manipulations now is experimental because we don't know what the outcome is going to be of actually changing those DNA sequences in the diseased individual. So yeah, a long rambling consideration to your point, I think, Rick. I don't know if that got an answer in there.
I have a question. Andrew, can you hear me?
Yes, I can hear you.
Oh, okay. About 18 years ago, I had stem cell therapy on my knee. I couldn't walk hard. In those days, I could hardly walk at all. And what they did is they extracted stem cell rich blood and mixed it with a shrimp skin concentration, which I assume is the protein carrier to get it back into the body. You can tell me if that's true or not. But anyway, I got 35% growth of the meniscus over the years. So is that kind of what CRISPR would do to is it change something in my body that has allowed for that growth of the meniscus?
Yes, yeah. So probably, and I don't know the specifics of the system you're describing, but I can make a sort of informed guess about what was done in your case, where those stem cells were taken. And so stem cells are attractive for correcting all sorts of disorders, including genetic ones like we've been discussing, because what they are only semi-differentiated cells. They're cells that maybe slightly committed to becoming a certain set of tissues, but they are ready to, once they get the right signals from the body, develop into the appropriate tissue. So by manipulating stem cells and putting them back in a person's body, the stem cells that we have manipulated, and in your case, did you say a shrimp protein, is able to regrow and maybe correct something like damaged tissue. And those types of treatments have been used for a long period of time. And in some of them, certainly there has been genetic modification of those stem cells. It's just that the old ways of modifying the genes were quite laborious and quite inaccurate a lot of the time. And, you know, inaccuracy might not seem like such a big deal if you're just taking some stem cells and injecting them in the knee, because it's true, if you cause a lot of unintended mutations, most of them probably will have zero effect on your knee or your overall health. But anytime you cause unintended mutations, there's also the risk of very severe disease like cancer, right? So cancer, which will ultimately impact every single human if they live long enough, is really an accumulation of mutations that cause different dysfunctions in cell processes. So that's one reason to be very concerned about having unintended mutations. And the old-fashioned ways of manipulating stem cells had higher risks of those unintended mutations. Still fairly low risk that you're going to create cancerous stem cells in your knee, but that's one of the reasons that we need to get it very right if we're going to go editing genes.
Gotcha, thanks.
I see it is
the code breaker, which I have not read, but thanks for the recommendation. Sorry, Bev.
Oh, I was just commenting on that. That sounds very interesting. I think I'd like to get that. Yes. Is there anything else? Does anyone else have a question? Have we answered everything? Is everyone good?
Okay, well, thanks everyone for your patience with all the technical challenges. I'm glad we made it work.
Andrew. Yes, thanks so much, Andrew. This was so interesting. I'm gobsmacked, really. It's just very, very interesting. Thank you so much for taking the time.
Thank you.
Yeah, most welcome. Thanks so much.